Nitella. Nitella species is a Caracean algae or stonewort. Just like Chara, Protocara, Nitellopsis, Tolipella, all these Caracean algae, they prefer shallow water. That is, they occupy the lake, the bordering of the lake, or any other water body, the shallow zone surrounding the water bodies. And they can be found in both fresh as well as brackish water, but the water should be clean. It should not be muddy. As Nitella prefer to thrive, they thrive well at a temperature between 20 to 30 degrees C. So, this species usually, though it is cosmopolitan in distribution, but in the tropical region, this Nitella, they disappear during the summer months when the temperature is very high, and in the temperate region where the temperature is very low during the winter month, then also this Nitella species disappear during the winter month in the temperate region. So basically this Nitella, as it is growing in the shallow water, it is a food and shelter for a wide variety of aquatic species. So very important plant. In India, we have many species of Nitella, as well as in worldwide also. It is cosmopolitan in distribution. Hello friends, I am Dr. Ayantika Das, and in this video I am going to discuss about an overview of Nitella, where I will be discussing about the distribution, morphology, anatomy, ecological economic importance, control measures, and some fossil forms of Nitella. I have also made other videos about other Caracean algae like Chara, and a link to that I have put in the description box. So let's begin the video. So overview of Nitella. Nitella belongs to division Chlorophyta. And what are the chlorophytes? What are their characteristics? That I have made a separate video. You can go to my channel and watch that video. It belongs to class Charophyceae. Now all the Charophycean algae, they are mostly aquatic, and they prefer shallow water. They are tolerant to salinity, and also they are tolerant to wide variation in the pH. So they belong to order Charales, family Characeae. This family Characeae is again divided into two subfamilies, Nitelloidae and Charoidae. Now Nitelloidae subfamily contains Nitella and Tolipella, and the Charoidae, they include Chara, Protocara, Nitellopsis, and many other such plants. Our topic of discussion today is Nitella, and we have got 37 species of Nitella, of which these are the few species that are very common. So this Nitella, along with Chara and many other Caracean algae, they have got this equisetoid habitat, which means that they have a jointed appearance, jointed appendage like appearance. We know that equisetum, it has got a jointed appearance, jointed segmental appearance. So this equisetoid habitat is very much common in all the Charophycean algae, and here the plant body is divided into a series of nodes and internodes, which gives a jointed appearance. So the appearance is a little bit different from that of the Nitella and that of the Chara, that I'll discuss when I discuss the morphology. So by habit, it has got a equisetoid habit, with we see that at the nodes there is a hole of lateral branches, and these lateral branches further they furcate. And about the habitat, it is found growing on water and submerged under water, both fresh as well as aquatic. But certain species like Nitella terrestris, it is a terrestrial species of Nitella, which prefer to grow in the damp soil, never grow under submerged condition. Distribution of Nitella. Nitella is more cosmopolitan in distribution than CHARA, but there are more endemic species of Nitella also than compared to CHARA. As they prefer to grow in a very short range of temperature, that is 20 to 30 degrees C, most of them they are found in the lower latitudes, that is in the lower tropical region and the temperate region, but Nitella flexilis is found in the temperate region also. There is another species, Nitella capillaris. They are found in in the temperate region, that is Europe and North American region. Apart from that, there are also many species like Gracilis, Hyalina, Mucronata. They are cosmopolitan in distribution. So here we are finding Nitella flexilis, and here we are finding Nitella capillaris. So Nitella capillaris and flexilis. In India, we find Nitella anamylaensis in Tamil Nadu, and in West Bengal we find Nitella flagelliformis. So these two species of Nitella are found in India, and in India this Nitella anamylaensis has been discovered quite a long time ago by Sundarlingam et al. Morphology of Nitella, that is the external appearance. Now, just like other Caracean algae, Nitella is also erect, slender and long, and has got a equisetoid habit, which means that the erect algae will have a jointed appearance, that is a succession of nodes and internodes, and at the nodes will be borne a whole of lateral branchlets or leaves of limited growth, and generally this whole will contain 4 to 7 number of branchlets or leaves. From the axis of these branchlet and or leaves arises a branch of unlimited growth that are also called auxiliary branches, because they arise from the axis of the branchlets. Now these branchlets in Nitella, they are, as you see, this equisetoid habit, and these are the branchlets. So, and at very base, at the base, basal nodes will bear, the basal nodes of the Nitella will bear a branchlet or a branch of unlimited growth, also called the axial branch. So this is Nitella mucronata, it's a very common Nitella. So we see that they are born at the nodal region, whole of branchlets and axial branch, they are born at the nodes. And one thing, despite the equisetoid habit, Nitella is different from that of CHARA by having ecortication. That is, the in just like CHARA also has a succession of node and internode. Nodes will contain the CHARA nodes will contain 2 cells in the center and 6 to 20 peripheral cells, but in case of this is the case of CHARA, but in case of Nitella, it is 2 central cell and 4 to 6 peripheral cells at the node. But the main difference, not only in the node but also in the internode, the Nitella, it does not have any cortication, which means that there will be a single elongated internodal cell. So this is the node and this is the internode. So basically this cell, this is the internodal cell, this one. This is the internodal cell, and this is a single internodal cell that is much elongated, and this single cell is not covered by any other investment of cells, unlike that of CHARA. In CHARA, the single the central cell has a cortex of many 6 to 20 cells, but that is not the case. That is not the case in case of Nitella. So this is basically the external morphology. One thing that from the basal nodes, that is from these region, that is the basal nodal region, from the nodes at the very base of the plant arises rhizoids. Now these rhizoids help in attachment of the Nitella to the muddy or sandy substratum. So these rhizoids, they are basically multicellular, but the septation between cell is incomplete. They have an oblique septa, and they are branched, and they are colorless. So we see in the morphology of Nitella, it is an erect branched plant algae with with whole of branchlets and axillary branches in the nodal region of the stem, and from the basal nodal region there arises rhizoids. And the sex organ, they are born on the branchlets, and we know that the branchlets, they are forked. Here we see that if this is the central axis and we have a hole of branchlets, and each of these branchlets are again, they are forked, and at this forking the sex organ develops. That we'll discuss later. So the anatomy of the nodal cell, we know that if this is the main axis of the Nitella, then they will have nodal cells, nodal region of 2 central cell and many peripheral cells. Let's draw only 6, because otherwise it will become very clumsy. So this is the nodal region. So and this is the internodal region. So this is the internodal region, and this is the nodal region. So the cells in the nodal and internodal region, though at a young stage they are similar in appearance, but at maturity this internodal cell, they become quite different. So here I am going to discuss about the morphology of the nodal, of the anatomy of the nodal cell. Now if this is a nodal cell, this is a very rough drawing. So nodal cell will have a cell wall. So let's say this is the very rough drawing. So this is the cell wall. So the cell wall of the nodal cell will be cellulosic and external to the cell wall. So if this is the cell wall that I am writing as CW, so external to the cell wall, there will be a covering of mucilage. So external to the cell wall will be a mucilaginous covering, and in fact this mucilaginous covering on the surface of the Nitella cell, many periphytons are found to be growing. So this is the mucilage, this that I am drawing in yellow. Then inner to the cell wall is the plasma membrane, very adjacent to it, the plasma membrane that I am drawing in green. So the plasma membrane, inner to the plasma membrane is the cytoplasm, and in the nodal cell the cytoplasm is very dense, and we can see cyclosis of the cytoplasm. But so I am drawing this one. This is the plasma membrane that I am drawing in green. So inner to the plasma membrane is the cytoplasm, which contains various organelles. For example, they have got a centrally placed large nucleus. Only the nodal cell will have a centrally placed large nucleus, and then this is the nucleus that and it will also contain many other organelles, for example discoid chloroplast. One thing that is common in all the charophytes are that the discoid chloroplast will not contain pyrenoids. There will be many thylakoids stacked together to form this chloroplast, but pyrenoids that are totally absent. Starch grains are stored, but pyrenoids are absent. So these are basically the plastids, the chloroplast. And then it has also got many mitochondria, it has got dictyosomes, it has got spirosomes, and also some small amount of vacuoles. The vacuoles are the internodal cell. Now at the very young stage, the nodal internodal cell, they have a similar structure, but as it matures, the internodal cell, it elongates, and then it develops a large central vacuole. So this is the large central vacuole that is formed inside the internodal cell and is characteristic of the internodal cell. Now the internodal cell has got an outer mucilaginous covering. So outer mucilaginous covering will be there, and next to the mucilaginous covering will be this is the cell wall, and internal to the cell wall will be the plasma membrane that I am drawing in red. So this is the plasma membrane, and then internal to the plasma membrane is the cytoplasm, and which is divided into ectoplasm and endoplasm depending upon the density. The outer dense cytoplasm, which contains, you see, which contains only the chloroplast that are aligned linearly, they are aligned. So this entire region from the plasma membrane to this region, this entire region is the ectoplasm, which contains, here we see that these are the chloroplasts that are lined. So the ectoplasm, though dense, it does not show any cytoplasmic streaming. Whatever cytoplasmic streaming is present, that occur in the internal region, that is in between this region. So this internal region will contain many nuclei. Earlier a single nucleus was present in the immature internodal cells, but the mature internodal cell will contain many nuclei that is formed by the division of the single nucleus. Now apart from nuclei, it contain mitochondria, dictyosomes, spirosomes, endoplasmic reticulum and starch grains, but this internal region will have cyclosis, that is streaming of the cytoplasm. There will be a descending movement, and then again it will move, and there will be an ascending movement, like here. This is the example, and this cytoplasmic streaming is very good characteristic and has been studied in both CHARA and Nitella to understand the mechanism of cytoplasmic streaming. It is more easier to study this type of plant physiological processes in algae. Because of their entire small size, they are easy to be cultured, and also they are very easily cultured with minimum requirement. Also they have a very simple body structure. That is why a single internodal cell can be just studied for this cytoplasmic streaming. So cytoplasmic streaming mainly occurs in this interface. In this interface between this ectoplasm and endoplasm, this interface that I am drawing here, this is the interface we see that these are the chloroplasts. These are the chloroplasts that are aligned end to end, and these are the microfibrils that are aligned here. So at the interface, this is the exoplasm and this is the endoplasm. So this is the endoplasm, that is the inner cytoplasm, endoplasm, and this is the exoplasm, that is EX, that I am writing in X. So in the interface region, we are having chloroplast and microfibrils, and this microfibrils, that is actin myosin, they generate the force in presence of ATP and magnesium ion. Motive force to generate the cyclosis is obtained in this microfibrils, that is the actin and myosin. They are responsible for the cytoplasmic streaming. So mainly this interface region between ecto and endoplasm is the primary source for generation of the force for cyclosis, and then it is transferred inward towards the endoplasm. So this is basically, this is a very good diagram which explains the cytoplasmic streaming. So this is the one thing I should be saying, that cytoplasmic streaming is also very much studied in CHARA species, also in the internodal cells. So both CHARA and Nitella, they are studied for cytoplasmic streaming. Now let's talk about the growth. We know that in CHARA and Nitella, all the Caracean algae, the main axis that shows indefinite growth, it is mainly due to the apical cell. This apical cell is a dome-shaped cell. This one, this dome-shaped apical cell now undergoes repeated division to form a series of cells. So if this is the apical cell, and it forms a series of cells just like a filament, and each of these segment, they divide into two cell. Each of this segment, they divide into two cell. This is the upper cell is the nodal initial, and the lower cell is the internodal initial. And the nodal initial and internodal initial, they also have differences in the wall thickening. And this nodal cell, what they do, they divide several times to form the central as well as the peripheral cell. But the internodal initial, they do not divide any further, they just elongate and expand. So this is basically how the apical cell forms a series of cells first, and each of these cells then redivide to produce the nodal and internodal initial, and then the nodal initial further divide, but the internodal initial do not divide, they just mature by expansion. So this is pretty much how the growth is apical, and it is limited to the main axis. And the now about the economic importance. Now we know that just like CHARA, Nitella being a Charophycean algae is of immense ecological, economic, research importance. And Nitella flexilis and Nitella furcata, these are the two they are the two species of Nitella that are used as a model organism for studying various plant physiological process. And this is because first is that they have got the internodal cells are much larger in size, and they are also easily cultivated, and they are used for cytoplasmic streaming, for the study of cell wall synthesis and many other studies. And in fact, they are also used for studying the cellular organization. So this is their use in understanding the plant physiological process. So this is a lab use. Now apart from that, it has got economic importance also, because it is a food for both aquatic invertebrates as well as small fishes, as well as waterfowl. Apart from its use as a food, it is also a shelter for small fries of fishes as well as aquatic insects. So this is both food as well as refuge or shelter for the aquatic organisms. Apart from that, it also stabilizes the mud. It stabilizes the sediment, and so any area that is very has got a clear water, you are bound to get some amount of either Nitella, CHARA, or this type of myriophyllum that stabilizes the basic mud. So it is basically also stabilizing the lake sediments. But one thing is that it is also, these are the negative impacts. These are the negative impacts, that is, when there is a little bit of eutrophication, then it will grow excessively, and then it will cause choking of the irrigation canal, or it will hamper the recreational activity. Excessive growth of Nitella can also be the breeding ground of mosquito, because apart from other type of invertebrates, aquatic invertebrates, mosquitoes also find refuge in this CHARA and Nitella that is found submerged. So this is a very important, because though CHARA and Nitella might look similar, they have got a jointed appearance. But if we look at it very closely, then we will find this one is the CHARA and this one is the Nitella. Though they have got a jointed appearance, but there is a basic morphological as well as physiological difference. The main morphological difference we see that though the CHARA is also has got a whole of primary laterals, and Nitella has also got primary laterals. But the primary laterals of CHARA, they are not itself branched. I mean, the primary laterals, if this is the stem, main axis, so primary lateral, this is a primary lateral, it can in turn sometime bear the secondary lateral. But in Nitella, if this is the main axis, each of these secondary laterals will be either twice or thrice or even 4 times, 3 to 4 times furcated. So there will be a furcation, and it will give a much more bushy appearance to that of the Nitella. So the Nitella has got a much bushy appearance. Apart from that also, the growing condition, Nitella, it prefers more acidic water, and CHARA can tolerate alkaline water. So Nitella is preferring more fresh water than CHARA, and also there is a basic general difference that we can see by touching. This CHARA has got lime encrustation, and so it is the Nitella do not have any lime encrustation. That is why Nitella is soft to touch and CHARA is hard and gritty to touch. And apart from that, CHARA, when crushed, it gives a very garlicky odor, musty odor. That is absent in Nitella. So just by having a field study, we can understand whether it is in CHARA or Nitella. Also, there is certain difference, that is the Nitella is translucent green, whereas CHARA is greyish green. And Nitella, though it can grow in shallow water, but Nitella can also grow in deeper water. But in case of CHARA, we find them growing only in shallow water. And also the number of species of CHARA in India is much less than that of the. Apart from that also, if we see the anatomy, we see that in the CHARA, this one is the Nitella, and this one is the CHARA. So we see that the internodal region, this region, there is cortication. That is, a single internodal cell is covered, is ensheathed, or having an investment of many other cortical cells. This is called the cortication, that is found in CHARA. But in Nitella, this entire internodal cell, this is a single cell. There is no cortication. So cortications are absent in Nitella, and the Nitella, the primary branches, they are furcated, whereas in CHARA, the primary branches, they give off secondary branches at nodes. So that this is a hold appearance. This is a hold appearance, which means that if this is the secondary lateral, if this is the secondary lateral and it is again divided, so in each of these nodes developed, there will be a hold of secondary laterals. So this will be a hold structure, that is in CHARA, the primary laterals again they have a hold appearance, but in Nitella, the primary laterals or the branchlets, they have a furcated thing. This is a furcation. That is, this is a furcation where we see that it is furcated 3 times. Again, there can be more furcation here. So we see that basically the primary laterals also have a difference apart from internode, and also we see that the number of nodal cell is also bit different. Here this is the nodal arrangement in CHARA, and Nitella, and this is in CHARA. So we see that basically there is also both external and internally CHARA and Nitella are different. So the control measure, as we know that among all the charophytes, CHARA and Nitella, they are very much tolerant to eutrophication. In fact, they grow excessively in case of eutrophication. So we should be very careful in whatever we are putting in our water body. There should not be any runoff from agricultural land that will contribute to eutrophication, because once there is eutrophication, then we have to again do this mechanical control, chemical control, and biological control. So we can just prevent the agricultural runoff or urban runoff from falling in the water bodies. Then only that is the best prevention measure. Apart from that, we can have also control measure, like very tiresome, like mechanically raking it out, pulling or cutting, or we put a screen mesh, that is a burl of fiber or a screen mesh that will not allow the light, the much light to pass to the bottom of the pond, but that again will cause total loss or very stagnated growth of other type of aquatic plants. So meshing or covering the surface of water body, it is not that much good approach, because it will affect other type of submerged plant also. There can be chemical control. Now chemical control, there are many herbicides. These are the name of a few herbicides that are very common, but in that case also you should be very careful, because we should not apply herbicides to the entire pond at a time. It will be just 1/3 of the pond herbicide is applied. Again, after wait of 1 or 2 weeks, another 1/3 is applied, and in this way we will do it phase by phase. And also there are certain chemical herbicides that are not to be applied by us. There are certain agencies that that are responsible for providing this herbicide. They will be doing the entire chemical control, and that type of chemicals, they are not actually sold to public. There are certain recognized agencies that will be doing the entire chemical control in your water body. So that chemical control approach, there is so much restriction, because this chemical control not only affects the CHARA, but also affects other type of herbs as well as aquatic plants and animals. So this will in turn pass on to the food chain. So the chemical approach is not that much advisable, because they will in turn enter our food chain. The mechanical approach is also very time consuming. This one, mechanical approach is a very time consuming, very labor inefficient process. But the chemical control is also equally time consuming, but not that much labor inefficient. This is very much labor efficient, but then it is not ecologically fit chemical control. So then we have this biological control, which is not only very labor efficient, very time efficient, but also it is very ecologically also very important, because for controlling CHARA or Nitella, we can use this triploid grass carp. And we know that most of the polyploid plants and animals, they are generally sterile, because at the time of gamete formation, there will be certain there will be not as they are triploid or any other polyploidy, so there will be not equal distribution of chromosome. So some chromosome can form during the anaphase stage, the separation will not be complete. So some gametes will contain more number of chromosome, some gametes will contain less number of chromosomes, and so in this way they will have a heterogeneity in the gametes, and this may lead to sterility. So that is why we are using this polyploid grass carp, this fish, this carp. They mainly we are using this polyploid because they will normally feed on this Nitella or CHARA, but they will not be able to reproduce. So we do not have to control the biological control organism that we are using. We do not have to control it. It will stay in the water body as long as it leaves, and then it will have a lifelong control of these type of invasive type of algae. Fossil forms of Nitella. Paleo Nitella has been found in Catalonia of Spain, as well as Rhine-Chert in UK, in the belonging to the Lower Cretaceous period. And this Paleo Nitella, though we find this fossils of Nitella are quite rare than that of CHARA, because CHARA, they have an incrustation, lime incrustation in their reproductive organ, especially the female reproductive organ after fertilization is getting encrusted with a high amount of lime. But in Nitella, that amount of incrustation is absent. That is why we can find widely we can find gyargonites rather than the cortoids. Now, cortoids, they are the female reproductive organ, fossilized female reproductive organ of Nitella, whereas gyargonites, they are the female reproductive organ of CHARA, that is fossilized. And they both this gyargonites and as well as the cortoid or cortolites, both they are found in the limestone sedimentary rock, that is the micritic limestone area. Now this is the picture of a micritic limestone that has been found in the Catalonia region of Spain. And this here the this small spots, they are the cortoids or the female reproductive organ of Nitella. Along with this Nitella, the female reproductive organ of CHARA was also found, along with many other foraminifera, and that is the single cellular animals that contain lime in their external shell. So these three type of organism, that is the CHARA, Nitella, these two charophytes, were found in the same region along with many foraminiferas. And so this is the limestone micrite that has been found from Spain, and this picture has been taken from this paper of these scientists. So you can easily go to their go to Google Scholar and get more information about this fossil if you like. So these are some few more picture, and you can see that this is the female, this is the fertilized egg cell, and these are the tube cells. We know we will be discussing about the reproductive organ in the part 2 of this video, that is Nitella. So for now, this is the transverse section where this is the egg cell or fertilized egg, or that is the oospore, and these are all the tube cells that surround the central egg cell. So this is how it looks. This is the longitudinal section. This one is the longitudinal section. This is the egg cell or the ovum or the oospore, whatever we say. So, and these are the tube cells that are spirally arranged, we can see. So this picture also taken from the same paper, and this entire fossil, they were found to be belonging to the lower Cretaceous period. This is again another the thallus organization. This is the internodal cell with a hole of branchlets here. So we see that it is pretty much well preserved, this cortolite. And in India also, Nitellites sahani was found, but later it was considered very dubious. This was found in the Rajmahal series, that is in the Bihar Jharkhand area. So that was the first part of the video where I discussed about the morphology, anatomy, distribution, ecologic, economic importance, similarity of Nitella with CHARA, similarity and dissimilarity, and also the biological control measures. So in the next part of the video, I'm going to discuss about the reproduction and life cycle. So that video will follow this video. So I'll be soon coming with that video also. If you like my video, do hit the like button and do share it with your friends, because that will improve my views, and do subscribe because your subscription, it encourages me. And those who have already subscribed, thanks for subscription. And if you have any doubts or you want me to make any new video on any topic, do write it in the comment section. I'll definitely make that video of your request. Thank you again for watching this video.